Let's start with a simple but powerful idea. Analytical methods are not just lab procedures. They are the backbone of product quality. In fact, many regulatory delays don't happen because the product is bad. They happen because the analytical method isn't strong enough. Regulators today don't just check results. They assess the entire life cycle of the method, from development to validation to routine use. So in this video, I'll walk you through a practical checklist to build analytical methods that are not just scientifically sound, but also fully ready for GMP and regulatory submission. Before you begin method development, you need absolute clarity on one thing. What is the purpose of this method? Are you quantifying the API using an assay method? Detecting trace impurities, as per ICH Q3A and Q3B? Or evaluating dissolution to predict in vivo performance? Maybe it's a stability indicating method to track degradation over time. Each of these requires a completely different approach, different sensitivity, different validation criteria. So the first checkpoint is simple. Define the purpose clearly, because everything else depends on it. A strong method always starts with a deep understanding of the molecule. You need to look at key physicochemical properties like pKa and solubility, because these directly influence retention behavior and method design. Then comes forced degradation, exposing the drug to acid, base, heat, to understand how it breaks down. And don't ignore photostability. Light sensitivity can impact both method performance and packaging decisions. The more you understand your molecule, the fewer surprises you'll face during validation or regulatory review. Now we move into method development, and this should always be structured, not trial and error. First, select the right technique, whether it's UV-Vis, mass spectrometry, or ELSD, based on sensitivity and requirement. Next, define the chemistry. Choose the right column, like C18 or HILIC, and optimize mobile phase pH. Then comes optimization, adjusting flow rate, temperature, and gradient conditions to achieve proper separation. And one critical habit, document everything. Every change, every trial should be recorded clearly. Because during audits, your scientific reasoning matters just as much as your final result. If there's one parameter that can make or break your method, it's specificity. Your method must clearly separate the API from impurities, degradation products, and excipients. Chromatographic separation should be clean and well resolved. But that's not enough. You also need peak purity assessment, ensuring no coelution using spectral comparison. And most importantly, forced degradation should prove that all degradation products form distinct, resolvable peaks. Because if your method cannot distinguish between components, it simply won't pass regulatory expectations. Validation is often seen as a checklist, but in reality, it's proof of reliability. Let's break it down. Accuracy ensures your method measures the true value, typically checked across 80, 100, and 120% levels. Precision confirms consistency, with repeatability and intermediate precision, usually within 2% RSD. Linearity shows that your response is proportional, with an R-squared value of at least 0.999. And sensitivity is defined through LOD and LOQ, using signal-to-noise ratios of 3 and 10. Together, these parameters demonstrate that your method is not just working, but working reliably under defined conditions. Now comes the real-world challenge, robustness. Because a method that works only under perfect conditions is not a practical method. You deliberately vary parameters like pH by plus or minus 0.2, flow rate by 0.1, and temperature by about 5 degrees. The goal is to simulate small variations that happen across labs, instruments, or analysts. And your method should still perform, without impacting resolution or peak symmetry. Robustness is what ensures your method survives beyond development into routine use. At this stage, your focus shifts to documentation and submission readiness. Your analytical method must align with CTD Module 3 requirements, along with ICH Q6A and Q14 guidelines. You need clear GMP SOPs that describe step-by-step execution. Validation reports must include traceable raw data. And data integrity is critical, with secure audit trails and complete transparency. Because for regulators, well-documented science builds confidence in your product. Let's quickly bring everything together into a practical checklist. In the design phase, ensure the purpose is defined, the molecule is well understood, and forced degradation is completed. In the verification phase, confirm specificity, complete full ICH validation, and test robustness. And for submission, make sure GMP SOPs are ready, CTD Module 3 is aligned, and data integrity is fully maintained. This structured approach minimizes risk and makes your submission much smoother. I'll leave you with one final thought. The best analytical method is not the most complex one. It's the one that is right, repeatable, and defensible. If your method can consistently deliver accurate results, withstand real-world variation, and stand up to regulatory scrutiny, then you've done your job well. And that's what truly defines a successful analytical method. Thank you for watching.